I do like, I don't know, kind of want to just say goodbye, I guess, because it's kind of, this class is so weird because, you know, the next time like Thursday, seniors are going, a lot of the seniors, most of them are going to their like internship programs and then they graduate. And so it's kind of, for a lot of you, it's the last time I'll get to see you. Um, we are going to be playing some games for most of it, for especially the juniors and stuff. So looking forward to that. And like, I don't know, I'm so torn like with how I feel right now. Because on the one hand, I'm like, man, I can't wait to just play board games, and I have to like actually like I've basically been a student this year. I don't know if you know this, but like I really didn't understand a lot of what I taught you this year until like a few days before. Not like I've never learned it, but it's kind of like a lot of review, kind of like actually read your book and like, oh wow, that's how that works for some things. And so I'm kind of looking forward to not having to like prep as much, but at the same time, I don't know, it's like this will never happen again. Like kind of this collection of students, and it reminds me of like how you might feel when you watch like a TV show that's like Friends or something. You get to like the end of Friends, and you're like, well, guess I'm gonna go to bed now. Kind of like you don't know how to feel, right? You're like you had so many great memories from like that show and stuff, but then it's just over. And in a much deeper way for you because you all actually are real people and stuff. I don't know. I feel like kind of a lot of good memories and different conversations and stuff that I really value. Like, I remember a few years ago, like my best friend picked me up from the airport. Went to visit him, and he had this super nice truck. Like, it was literally like the nicest truck you could possibly buy. It was like disgustingly nice for like a 28-year-old, and I was like, man, and he's doing really well doing like roofing sales, like selling a lot of trucks and stuff, and that was kind of like that was sort of his reward for doing well at his job, and and I was kind of thinking like, man, I don't think I'll be able to like afford something like this, and it kind of made me like think about like, well, what do I get for doing a good job, and for me like I get like the memories and stuff. I kind of get like every year I get something like 160 something students that all a little bit different, but I kind of get to I don't know, get to learn the different personalities of all of you and all that. So and that's really valuable to me. At the end of every year, I'll get a yearbook, and I'll go through and I'll like circle all the students that either I taught or maybe like interacted with kind of meaningfully in the hallway throughout the year. And then during the summer, like I'll go through and like I always kind of look through like all the old yearbooks and stuff, and just kind of just reflect on the memories and everything. But I don't know. I think the hard thing with being a teacher is I kind of feel like I kind of get to like plant seeds, if that makes sense. Like kind of, but I don't really get to see like the fruit of those seeds. It's kind of like I get to teach you and just sort of like hopefully make some kind of impact on your life, but then it's like you just sort of leave, and it's like, well, like what happened to that guy? Like really, like I'm just like going through like the old yearbooks, like my first yearbook, and it's like, oh well, man, I wonder what happened. And so what I'm getting at is I'd appreciate if you keep in touch every so often. Now, like I'm not saying like every 2 weeks, give me an update or something. I'm talking like once a decade, like just something like super like long term, just every so often. Like I'd appreciate like a little check-in. How you can do that, like I'll write my email up here. I mean, I don't have any plans of leaving McLean, but I don't know, life happens and stuff. So aj.blackford77@gmail.com. Again, it's like, oh, like for example, or you can even reach out on like social media or something if you want to. Although I'm never gonna like initiate social media stuff if like for those of you graduating. I remember when I graduated, I had a teacher that like I like wake up after graduation, I look at my phone, and this is when like everybody saw Facebook, and it was like friend request from like some like teacher, and it was like, man. And then I like accepted it, and I look at us, and he like literally my man like went through and like friend requested like all of his students. And I think it must have been like the notifications at like 1 a.m. or something, and I don't know, like I can understand if like there are things you post on social media that you're like, I don't know if I want Mr. Blackford seeing this, It's like, no offense, Mr. Blackford. So I'm not, I'm not gonna like necessarily initiate that, but it is kind of cool, kind of seeing like some old students, like when I first taught high school, like 6 years ago or whatever. It's like, oh, they have kids now, Or I had one student reach out, and this is actually kind of a cool story. I had like this guy who like I told him as a freshman, and like he would do nothing. He was one of those students where it's like you would look at him and be like, the thoughts might creep in, like I don't know if this guy's ever gonna do anything with his life. Like it was like really dark thoughts, but he like sent me a message on Snapchat, you know, and gave me and like let me know that he just finished community college and he was like getting like a degree in computer science and like had like secured this internship. It just blew me away. Like it really made me like appreciate that like, man, if that guy can have success, literally any student I've had can like have success. And so anyways, I know I'm starting to kind of ramble at this point, but keep in touch and yeah, no, it's been a great year. So thank you for all that. But with the AP exam tomorrow in mind, let's go over photosynthesis and cellular respiration. When big picture, understand the relationship between the two. That the products of photosynthesis, so organic molecules plus O2, when they say organic molecules, they're talking about glucose mainly. The reason why they don't say glucose is because technically from photosynthesis you get G3P, which then gets converted to glucose. But anyways, you get energy plus oxygen, and then a plant, or if we eat plants, plant or us will then use that glucose, put it into the mitochondria and do cellular respiration, where you then have to utilize oxygen. You have to use oxygen because oxygen is going to be an oxidizer that will then oxidize, break down that glucose into CO2 and H2O, and the energy stored in the chemical bonds of glucose will then be converted into ATP. Okay. One last thing that I forgot to mention. When I had a few different AP teachers do this. Like the first day of school, they showed a graph of their like AP scores, and it was like my scores, the other guys, and it was like, and it was really more like the point of it was like to get buy-in, like, hey, trust what I'm doing, trust my system, I get results and everything, and I'm not gonna make a graph like that. And I bring that up because, like, I worry for some of you, like, if it doesn't go well tomorrow, if you get, like, a 2, it's like, well, better not show my face around that guy again or something, And I do care about, like, the AP scores, I more care just to, like, give me an idea, like, did I mess things up, It's like if Murdoch and Schultz are, like, up here and I'm, like, down here, it's like, man, I should probably really rethink my whole teaching career, and everything. But, yeah, just do the best you can tomorrow. It is what it is. One test is not going to define your biology career, whether or not you should question your life or something like that. Just learn from the mistake if it doesn't go well. And if it does, then congratulations. Anyways, there you go. So understand kind of that relationship there. And get the cursor over here. So, okay. Hard thing about cellular respiration is a lot of numbers, a lot of different processes. You do technically need to know the numbers. Technically, they could ask you how many ATP come from glycolysis and all the different other stages. Do the best you can with it, but the ultimate thing you really got to understand is big picture, what is happening. And what's happening is you're taking the energy stored in glucose, and that energy is stored in the chemical bonds of glucose. Well, what makes up a chemical bond? The sharing of electrons. So the electron transport chain, you're taking the energy stored in the bonds, the sharing of those electrons, and you're going to release that energy and use it to ultimately store it back into ATP. So then the cell, whenever the cell wants to use that energy, the cell can utilize energy. ATP, they're basically like little batteries cells can use. Okay? But we're going to do glycolysis. We're going to take glucose and turn it into 2-pyruvate. And then we'll do some, and when you do that process, you make 2 ATP. And you do this by substrate level phosphorylation. So this is done by an enzyme that will, like when they say substrate level phosphorylation, they're just saying like a substrate is what's used in the enzyme. There is an enzyme that will catalyze the formation of the ATP. But that's not the main point of glycolysis. The main point of glycolysis is not to make ATP. The main point of glycolysis is to make the NADH. And mainly, the biggest number one thing is the pyruvate. If they're going to ask you anything, highest priority will be asking about that pyruvate. So make sure you understand that. And understand that glucose, understand how to count carbons, okay? Glucose is a 6-carbon sugar. You're making 2 pyruvates because each pyruvate is a 3-carbon sugar, right? We can't lose the matter and stuff, so like you got to keep track of the carbons. When you're thinking of glycolysis, think 2s. You get 2 NADH. Think deuces. 2 ATP and 2 pyruvate. 2, 2, 2. Okay? So that is for glycolysis. And I think like that isn't that hard to remember. You think 2s, chunking up the deuce, doing this, whatever to get you to remember 2s. Okay? All right, so that's for glycolysis. Now then, so then that pyruvate is going to then enter the mitochondria. Also, you know the locations. Glycolysis is happening in the cytosol, otherwise known as the cytoplasm. We then put that pyruvate into the mitochondria, and before you do the citric acid cycle, there's a quick reaction that we do called pyruvate oxidation. Oxidation is referring to the removing of electrons from this pyruvate. That is why you're creating 2 NADH, and then you're going to make 2 acetyl-CoA. So again, keeping on the theme of 2s. You got 2 acetyl-CoA, 2 NADHs. 2, 2, 2. Okay? You also make 2 CO2s. So 2, 2, 2, 2. Okay? A lot of 2s going on. When in doubt, pick 2. Now then, that acetyl-CoA, and this is now, there's 2 of them, so counting carbons. Here, think of it as this. We lost -2 carbons. You're making 2 carbon sugars called acetyl-CoA. Those are going to go into the citric acid cycle. And really just understand what you're getting out of the citric acid cycle. You're going to get out 2 FADH2, 2 ATP, and 6 NADH. You are also going to get out 4 CO2. Now, if you notice my hesitation, it's because like I didn't have that memorized. How could I have figured out it was 4 CO2? What's that?
Is it minus 2 of CO2?
Are you saying because of this?
Yeah.
Yeah, exactly. Can you elaborate on that a little bit more?
So, this is so total there is 6 carbons, so you subtract 2 away 4.
Yeah, exactly. Yeah. So, like this, there is some common sense. These numbers aren't arbitrary, is what I'm trying to say. You start with a 6-carbon sugar. You know you're losing 2 CO2 before you go to the citric acid cycle. So you're like, well, I got 4 carbons, so that I must lose 4 CO2s. Okay, so if you kind of get a little confused in the numbers, just think of that. Here, I can write these numbers over here. So pyruvate oxidation, you got 2 NADH, 2 CO2, and then 2 acetyl-CoA. And this is pyruvate oxidation. Oh, and by the way, if you want to like do something else, like I don't care if you're like listening, like don't, if like at this point, like if you want to do some short answer stuff, if you feel good, that's fine with me. Don't mind me. I'm just doing this just because maybe it'll help some people. But anyways, pyruvate oxidation. Then you have citric acid cycle. So citric acid cycle, you're making 2 FADH2. So FADH2, it acts like NADH. They're electron carriers. They're going to carry, they store energy from that the electrons that were stored in the glucose, and they're bringing us to the electron transport chain. So FADH2, you make less of it than the NADH, but they act in a similar way. So 2 FADH2, 2 ATP, 4 CO2, 6 NADH. So 2246. 2246. So 2222222222246. There you go. And this would be the citric acid cycle. Citric acid. Okay. And again, the ATP here, that's also made in the citric acid cycle. That's made by the substrate level phosphorylation. So an enzyme's doing that. The location of these, they'll say it's the stroma. It's basically like the cytoplasm of the, or I'm sorry, not the stroma. That's the chloroplast. The matrix of the mitochondria. Okay. All right. So then, now we got all those electron carriers. They are then going to be shuttled to the membrane area of the mitochondria, where we're going to be doing what's called oxidative phosphorylation. So I want to move on from this slide, 'cause it's kind of hard to explain this process from here, but I'll keep this other stuff up on the board. A little bit more on glycolysis. Understand that there's 10 different reactions. If you remember, that's like a long list of reactions. You don't have to memorize those reactions until you get to, if you do like biochemistry in college, they make you memorize those structures and stuff. All for your purposes, just understand that reactions 1 through 5, they call that the energy investment phase. You basically kind of have to like prime the pump. You gotta, if you're trying to roll a boulder over like down a mountain or something, sometimes you gotta get it up to the very top of that hill first. That's the energy investment phase. And then you have the energy payoff phase. So understand energy investment, you're gonna invest 2 ATP and then you get out 4 ATP. But net get out is 2 ATP. Okay, so they might try and play games with you on that, but make sure you don't let that confuse you. It is 2 ATP, but it's 2 net ATP. Here's a citric acid cycle. I'm not really going to linger on it much longer than I already did on the last slide. Really, what they would ask you about it is just inputs and outputs. Really, this right here. If you understand. Which, to be sure, these are the outputs that are coming through. So understand that acetyl CoA, that's going to be going in to the citric acid cycle. There you go. So then now the electron transport chain. So this is more of a process to understand. So this isn't so much like things to memorize. This is something you should be able to describe. Like, be able to describe what's happening here. And it seems really complicated, but I really think it's kind of like Where's Waldo? When you see Waldo, you just see him. It's hard not to see him. When you see it and it makes sense, I think it can stick with you pretty well, because it's just a system. It's a cool thing to like actually understand, and you can memorize it just by understanding and not by like literally memorizing numbers and stuff. Anyways, understand that the NADH and the FADH2, those represent stored energy. Stored energy in the form of those electrons that we at first removed from our glucose. We are then going to, what we're doing is we're taking those electrons and we're going to pass them down these protein complexes. So when you see 1, 2, 3, 4, these are just a collection of a bunch of different proteins, and their job is to remove the electrons from these stored electron carriers, and they're then going to create this hydrogen ion gradient. So you're going to end up getting a high concentration of H plus in the intermembrane space. So understand the structure of the mitochondria. The mitochondria has 2 lipid bilayers. So what they're not showing here is you have another lipid bilayer up here that would stretch all the way across here. So the intermembrane space is the space between the outer lipid bilayer and then the inner lipid bilayer, the intermembrane. Okay? Intermembrane space, understand it's going to have a high concentration of H+. Now, what's its pH? High or low? If you have a high concentration of H+. Low. Okay, so we didn't really touch too much on pH, really, if at all. Understand that high H+ is low pH. They're backwards. Okay? All right, so you get all those H+. Now, the source of those H+ is from the H on NADH and FADH2. I didn't really mention this in detail, but NADH was at one point NAD+, and then it turns into NADH when you get those electrons from the glucose. Okay, so make sure that makes sense to you. Anyways, so you got all these hydrogen ions going up there, and you're like, well, what's the point of that? Like, what does that do for us? Well, how does that actually lead to ATP? Well, thinking back to diffusion, where diffusion things want to go from more to less, from a high concentration to a low concentration. So the point of this concentration gradient is you want to do something called chemiosmosis. So osmosis, referring to, when we say osmosis, we're specifically referring to the flow of water. but like I think it's Greek. The original language, osmosis, is really just referring to like the pushing of something. It doesn't have to be water necessarily. So these hydrogen ions want to go from a high concentration up here to a low concentration. So understand there's a low concentration. So a higher pH in the mitochondrial matrix. This is the matrix of the mitochondria where we did the citric acid cycle. So those hydrogen ions are going to go through the ATP synthase, and it functions like a wind turbine. I was telling you this is like the world's smallest turbine, where the idea of a wind turbine is the wind is going to move the turbine, and in the movement of that turbine, there's a generator inside that wind turbine that will then create an electric current. There's not a literal generator in here, but in a similar idea, there's enzymes at the very bottom part of this ATP synthase that the movement provided by the H plus will spin this like motor type thing in there. It's made of proteins, and will catalyze the reaction where you will add a phosphate group to ADP, making ATP. But again, the powering of that comes from the H plus, which came from the NADH and the FADH. Okay, and then that will make this makes the majority of your ATP. Think somewhere around 26 ATP. They're not going to get super specific about that number because that number will change depending on just different variables. They're not going to be as like strict as they are about these numbers. Okay, something around 26, maybe 28, 24. I doubt they'll ask you specifically, but do know that this is where you get the majority of the ATP. That's really the important thing to understand. Bless you. Now, this whole process, so when you include the electron transport chain and then chemiosmosis, that whole thing is called oxidative phosphorylation. So I'm going to write that a little bit bigger because I think it's worth breaking down that word. Oxidative, we are oxidizing. What are we oxidizing? That's an important thing. What are you oxidizing, Thomas? NADH and then to a smaller degree the FADH2 to then phosphorylate the ADP to make ATP. Okay. All right. Questions about cellular respiration? I know it's a lot, but I don't know. I think it's really it's worth just kind of memorizing and stuff. I think you can get a lot of easy points from this. So, all right, I want to talk about before I talk about photosynthesis, I'm going to talk about essentially getting into this idea of like, well, what happens if you because cellular respiration. Oh, I forgot to mention this. Oxygen. So they have O2 down here. The reason why it's cellular respiration, you need oxygen. Oxygen is the final electron acceptor. I'm going to say that again because it's, I don't know, I feel like I had that as a test question like 3 different times throughout like my college career. People like asking that. Oxygen is the final electron acceptor. Why is it the final electron? What about oxygen? Like, thinking chemistry-wise makes it like a really good, yeah.
It's very electronegative.
Yeah, it's really, really electronegative. You remember like the trend of electronegativity? You trend towards the top right of the periodic table. Oxygen is super electronegative. Anyways, and then the oxygen will combine with some H pluses that are still in the matrix to form your H2O. So technically, you make water as a product of cellular respiration, but it's technically really a waste product. It's not really it's not an actual goal to make water in cellular respiration. Okay, so then, you need oxygen to do all of those processes. So understand that. If you want to do all of cellular respiration, you have to have oxygen. If you don't have oxygen, you can only do glycolysis. Okay? So glycolysis does not need oxygen. Okay? So if you're this is where you get into like ethanol and lactate and fermentation, lactic acid formation. This is where like you get like the lactic acid, like the class thing like with like runners or people like different sports athletes and stuff where they're pushing their body beyond like their oxygen capacity, but they still need energy, so they would just do glycolysis to make just that 2 ATP, which isn't much, but it's better than making no ATP. But then, it's like it's like surviving off of like running credit cards and stuff. You know, there's going to be an interest fee associated with that. That interest fee is the lactic acid or the ethanol, which are waste products. They're toxic for the body. Okay. So, talking about fermentation. Fermentation, again, you're doing glycolysis, where as we talked about glycolysis, you make 2 ATP and you make 2 NADH, and you make 2 pyruvate. Now, what's interesting is the main point of cellular respiration really was to make those NADHs. But here, the NADHs are actually they're of no use to us. Why? What did it say? Exactly, right. You can't remove those stored electrons because you don't have oxygen as that final acceptor of the electrons. Okay. Anyway, so then what's going to happen is you want that 2 ATP. That's your goal. That's why we did this in the first place. But in order to do glycolysis, you have to have NAD+. So we have to do something called NAD+ regeneration. So whether you're talking about fermentation or lactic acid formation, you're going to do the regeneration of NAD+. The difference between the two is how you regenerate the NAD+. In fermentation, you'll take the pyruvate, remove the CO2. This is why beer is carbonated. Thank God. Anyways, you remove that CO2, and then we can convert the acetaldehyde back into, so we'll take the electrons that are stored in NADH, and we're gonna put those electrons on this carbonyl group, turning it into an alcohol group. Hence, ethanol is an alcohol. Okay. This is a waste product. So this is how bacteria will do it, or like yeast and stuff. Like when they're doing fermentation. We don't do this process. So understand that. If you forget, we don't do this. We do something similar to this, called lactic acid fermentation. But before I move on, this NAD+, now we can then do another round of glycolysis. What we do, which looks super similar, is called lactic acid fermentation. So both are fermentation, but one is ethanol fermentation, the other is lactic acid. All right, so same idea. We need to make ATP, but we don't have oxygen. The issue is to make these 2 ATP, I gotta have NAD+. So to get that NAD+, I'm going to remove it. To regenerate the NAD+, I'm going to have to take my pyruvate and store the electrons from NAD+ onto the pyruvate so that I can then free up my NAD+ again. But then this will create something called lactate, or lactic acid, if it's lactic acid if you remove that hydrogen there. Anyways, not as important, but by doing that, you regenerate the NAD+, so you can keep doing glycolysis and making the 2 ATP. But again, this is a waste product. This is toxic to your body. This is why eventually your muscles just shut down on you. Like eventually you won't be able to just keep going, no matter how like tough you think you are up here. It's like your body will shut it down. You know, stop you from doing whatever it is you're doing. Okay. Last thing with cellular respiration is how they feed into the cycle. So different cellular respiration, it's focused on glucose, which is a sugar, right? That is your main energy source. But technically, like when you eat food, you don't just eat up straight just glucose, right? We eat not just sugar, we eat fats, we eat some protein, maybe some nucleic acids, and all of those biomolecules can feed into cellular respiration at different points in the cycle. You don't need to memorize where they feed in, but just understand that. Just kind of as a general understanding of like where they possibly could feed in. Like fats, they're made of glycerol and fatty acids. You need to know that, like what fats are made of. Glycerol, one of the products during citric acid cycle is glyceraldehyde 3-phosphate. The glycerol part actually comes from fats. So that's why you can't just eat straight up glucose, because we still need to get some of these other nutrients and stuff to build other things and everything. But anyways, that's kind of besides the point. Fatty acids can go in here at acetyl-CoA, and amino acids can be fed in at a bunch of different points. But then you'll release ammonia as a waste product, and that's why like your urine has a lot of ammonia in it, because of when you're breaking down those amino acids, you're going to make a lot of that NH3 that then gets put into your urine. Okay. Want me to keep going with photosynthesis? Or shit. Well, let's call it for now. I'll kind of feel it out, but I've been talking for like 30 minutes, and I'm gonna get through all photosynthesis. I might need another like 20 minutes, so maybe we'll just let it be. But reminder, like I do have those other videos of me reviewing photosynthesis. If you really wanted to hear me do it again, you could find that video.